recording going, and we're in business, so let's look at some jokes. I know this is the reason you guys all actually come to this. Has nothing to do with the test tomorrow. Pope says, Henry, you can't just go divorcing all these women, and he changes teams. So, me, can I go to the bathroom? Teacher, I don't know, can you? She's got me. I haven't evolved a standard animal digestive system. I continue to photosynthesize my energy. You guys hate photosynthesis, don't you? It's the reason you're alive, so... Alright, our biology fact for the day. Wild dolphins appear to create names for each other based on variations in their calls. So basically dolphins name each other and have like a specific call that they do when they are communicating with each other. And they've only figured this out over the last 5 years or so. I mean, it took more than the last 5 years or so. But about the last 5 years or so, they've realized dolphins will call each other by name, which I think is actually pretty dope. So they're going to take over the world someday. And they'd probably do a better job than we do. All right, my study tip is to study out loud, right? So rather than just reading in silence, okay, read out loud or say stuff to yourself out loud or explain a concept to yourself or preferably to somebody else, because then if you're wrong, they can tell you you're wrong, or if they need to ask further questions, then they can ask questions and it makes you need to understand it on a deeper level, right? So study out loud, force yourself to, I guess, learn in more than one way, right? Because not only do you read it and see it, but you say it and you hear it. And preferably you should draw it or write it if that's also possible, right? So like the life cycles that we talked about for the last 12 seconds of class, but that are going to be on the test, which you guys are super grateful for. Draw those, right? But then explain them out loud. Read from the textbook about them or read from the slides or watch the slides, but then say it out loud, okay? Saying it into the real world makes it a more real topic. All right, so wanted to talk about just some quick test-taking strategies before you guys go get into it, and I've talked about some of these before, but it's always good to get a refresher. Get pumped up, which I know sounds kind of lame because that's what you do for like sports, right? Right before a football game in the locker room, you play some loud music and you yell at each other and you hit each other because you're men, right? And you're tough. Okay, do the same thing for test taking, right? Maybe don't hit people or yell at each other. Guys, try and keep it down when you're coming in. Sorry. Talking mostly to the front right here. Thank you. Get pumped. Act like testing is a sport, right? Because for most of you, you probably don't even play a sport anymore if you ever did at all. So school is your sport now, right? And I know that sounds kind of lame and kind of dumb, but that's how I treat it, right? I treat each test like a big football game, a big soccer game where I got to get pumped and I got to be ready, right? If you act that way about tests, you'll do better, right? Because you're pushing yourself to perform. That's the whole point of a sport, right? And to get women. Eat a good meal, right? Eggs, toast, fruit. It says avoid sugar right after it says fruit. It means like don't have like a Dr. Pepper and a donut right before a test, right? Because you guys know how sugar works. We were all 5 once. You peak, and then 2 hours later, you don't want to move, right? It would be pretty bad luck if you crash during a test, right? You are far less likely to crash if you had eggs and toast and a glass of orange juice and whatever else they put on the table in a cereal commercial, right? Fruit and grapes, and those are fruit. Have a good breakfast. We'll put it simply. And then flag the hardest questions and come back, right? I talked about this in my super session, but it's always a good idea to flag the questions that you don't understand and come back to them, because a later question could actually help you answer an earlier question. Not always, but maybe you'll go through a question and you'll go, actually, you know what, that reminds me, this is the answer to that previous question. And since you've got it flagged, you can just go up real quick, find it, boom, you're done, right? So flag questions, okay? All right. This was his clicker question from class today. This seemed to trip up a few people. I've had a few people come up and ask me about it and email me about it because Chromosome number questions always seem to be the most difficult, right? Because it's hard to keep track. One chromosome's 2 molecules, but one molecule's 1 chromosome, and then they divide, and then who's where, and I don't even know what's happening, right? So let's take a second to look at this one. I simplified the wording because I didn't start typing it until he changed slides. It was like a fruit fly or something. Anyway, the diploid cell has 8 chromosomes. Undergoes meiosis 1. Now it has how many chromosomes? How many DNA molecules? Does anybody remember from class? B, right? 4 chromosomes, 8 DNA molecules. Who wants to tell me how they come to that answer? Raise a hand and be brave.
So it splits into, well, the 8 chromosomes, then split into 4 chromosomes, but there's still 2 sister chromatids. So there's 8 molecules.
So, a diploid cell, meaning, what does diploid mean? 2 what? 2 sets of chromosomes. Those are terrible. Anyway, there's 8 of them, right? I don't want to take the time. I don't even know if that's 8. Don't count them. It's 9. Okay, somebody walk me through it. What should I draw next? I'm not going to make this easy for you and just do it. I already know how to do it. Somebody else. Homologous pairs line up. Okay, that's a good next step. So we'll say those are homologous pairs. Okay. Did you mean like line up like in the metaphase plate, or did you mean just pair up? Okay, so they just pair up. Okay. Come on. Huh? Then they'll line up on the metaphase plate. Thanks for stealing that one. How long do you guys really want to spend on this question? Pull the pairs apart, right? So the pairs. That looks like a cartoon hand. There's a reason I'm a biology major, guys. Now what? There's a whole lot of mumbling and some hand gestures. So helpful. Cell divides. So I'm not going to draw another step, but I guess I should have 8 chromosomes so that if you were to look at this in reference to the question, you would understand what's going on. I don't even know what those are supposed to be. So how many chromosomes do I have in each cell? 1, 2, 3, 4. But how many DNA molecules do I have? 8, because each of those has 2 molecules, right? Now, had this said meiosis 2, that was awful, what would the answer be? A, 4 and 4, because now these would each divide in half. You'd still have 4 chromosomes, right? Because each of those molecules, when separate, is still its own chromosome, but it's only 1 DNA molecule. So I'll draw that because I saw some confused faces. So let's say this is one cell and this is another cell. We're going to look at just this one, right? So this one then splits in half again. So those chromosomes split in half, and it would end up, let's make sure I get the right numbers here, like that. And each of those dashes is a DNA molecule. But it's also a what? Chromosome. It's a chromosome. Okay, so did you see how meiosis 2, it would be 4 and 4. Meiosis 1, we haven't split the chromosomes, we've just separated the pairs. That's the kind of the big difference between 1 and 2. So that way it's 4 and 8. Okay? Question?
Yeah, so before all this happened, you just had a diploid cell containing 8 chromosomes. Would that mean that there's 16 molecules of DNA?
It's diploid, meaning there's two pairs of each.
Yeah, I'm saying.
So
in the I'm saying before it undergoes meiosis, would there be 16 DNA molecules?
Oh yeah, so if it said before meiosis 1 even starts, the correct answer would have been D, because you got 8 chromosomes, but you haven't done anything, you haven't separated them into separate cells or anything, so all 8 are in one cell, and since 8 chromosomes has 2 molecules? Yeah, it'd be 8 and 16 before meiosis 1, 4 and 8 after meiosis 1, 4 and 4 after meiosis 2. Make sense? All right, moving on. If you have questions about it, we can always talk about it later, but we have too much stuff to cover. So let's talk about some vocab first. This is a homologous pair of chromosomes. This is a homologous pair of chromosomes. Why? What does that mean, homologous pair? Same size, and they have the same genes in the same places. So if this one has that gene there and that gene there, this one will have the same genes in the same spots, right? And if this one has that gene there and that gene there, this one will have that gene there and that gene there, right? So their genes line up, which they have to be the same size for them to line up, because if one's shorter than the other, then they obviously can't line up, right? Parental and maternal. Paternal and maternal means just one from mom, one from dad. Well, I guess dad, mom, right? Sister chromatids, what are those? Replicated DNA, they're the mirror image, the identical halves of a chromosome with 2 DNA molecules, right? That's all the vocab for there. Let's see, so meiosis reduces chromosome number by half. Why take the time to point that out? Oh, wait. I'm not scared.
Because mitosis doesn't split in half. Mitosis only inserts chromosome numbers, so it's important to know the difference between that and like meiosis. Meiosis splits in half.
Yep, that's almost word for word what I was looking for. Mitosis doesn't, meiosis does. That's one of the biggest differences, right? That's why we care. Otherwise, this isn't any different, right? If it didn't reduce the chromosome number by half, it really wouldn't be all that different, okay? How does it do this? 2 back-to-back cell divisions without intervening chromosome duplication. What the heck does that mean?
The cell cycle doesn't run in between the 2 divisions. So
Perfect.
Replicate DNA in the S phase.
Yes, so we do S phase, but only once. So we do G1, S, G2, then meiosis, 1 and 2. There's not like meiosis 1, and then another G1, S, G2, and then a meiosis 2, because we would cut our chromosomes in half, but then double them again, and then cut them in half, and we'd end up with the same amount, right? So we do the whole cell cycle just once. Nobody comes in the middle and reduplicates DNA. It sounds like an arbitrary thing to bring up, but some people think that. Because they see meiosis 1, then meiosis 2, they think it's like we did the cell cycle and did meiosis 1, then we did the cell cycle and did meiosis 2. Not true. We do the cell cycle once, meiosis 1 and 2, and then we keep going. Okay. In animals, the haploid cells, what's a haploid cell? One set of chromosomes, right? So we're not talking about a 2N cell. We're talking about a N cell, right? One set of chromosomes. These are your sperm or egg. But we have 2 sets of chromosomes, so how does that make any sense? They combine, right? Sex. This is how it works. Sperm, egg. Each one brings a chromosome. Well, 23. And then you put them together and you get 46. That's how many we have, right? Questions there? No 8th grade questions. Real questions. No one thought that was funny? Fine. You guys. Somebody tell me what's going on here, besides saying it's meiosis. It's meiosis 1, you jerk. Care to expand? That's what I thought.
Is this the crossover?
We do have some crossing over going, but we'll get to that in a minute. It's part of it. Bigger picture. Crossing over does happen as part of meiosis 1.
The pairs, the homologous pairs are separated.
Yeah. So they've what?
They're separated.
At the bottom there? Yeah, okay, okay. Start at the top. Somebody start at the top for me.
Prepared to expand.
Huh?
I'm prepared to expand.
Do it.
Okay, so like you start with the chromosomes and then they duplicate.
Okay.
And then like you have like the homologous pairs and then they like
Okay.
cross over and then they split themselves.
Yeah, perfect. So we have our standard amount of DNA. It duplicates, right? We get a chromosome and a chromosome. They eventually line up. It doesn't show every single step, but you're going to undergo, prophase, metaphase, anaphase, and then down here, telophase. We send one whole chromosome with 2 DNA molecules and another whole chromosome. But see how there's like a little red bit and a little blue bit? They did some crossing over, but we'll get into that in detail in a second. And this cell splits, and they each go to one cell or the other, right? This is obviously with multiple chromosomes. We're just showing one, but this is meiosis 1. Okay? Question?
Why isn't this meiosis?
Meiosis 2 is coming up.
Because I thought in meiosis 1 you have like homologous pairs in each cell. So you have like 2 red ones.
I think it's just the picture here is as simplified as it gets. Meiosis 2 is when we split the homologous chromosomes in half. But you'll see that in a second. Does anybody? Yeah.
Do they have like certain steps that they're named, like prophase or?
Yeah, prophase, metaphase, anaphase, telophase. But
Still follows that?
it's still, yeah, so still the, what is that, PMAT?
Yeah.
PMAT, because there's a pro-metaphase. But in meiosis 1, it's prophase 1, prometaphase 1, anaphase 1, and so forth. And then in meiosis 2, it's anaphase 1 or anaphase 2 and so forth, right? So that they match up. So someone could ask you what happens in metaphase 2. You're going to have to know, first of all, they're talking about meiosis. Second of all, they're talking about meiosis 2, and then move from there, right? Other questions?
So do we contain like all of both of our parents' DNA?
Not all.
Like is this how we have? So the red ones, is that supposed to be like the maternal and the maternal ones?
I would assume so from this picture, yeah. What happens is your father, right, has his sperm cells, and they are haploid. They contain one set of each chromosome, right? Your mother, her eggs, also contain one set of each chromosome, totaling to 23 each. Then when they fuse, fertilization, they share them. But you don't contain all of their chromosomes in the sense that otherwise you would be exactly like both of them. You just contain the chromosomes that their eggs and sperm contain, and that's only one random egg and one random sperm. So it's a pretty small portion. Does that answer your question?
Yeah.
Okay.
So this is just like creating the haploid cells like the egg and sperm.
Yeah. That's the first half of the process right there.
So the red and the blue doesn't really differentiate chromosomes.
I'm guessing that yes. I don't know that it's specifically trying to say paternal-maternal in this case, because it's just, I don't know, just random cells, right? Okay. Other questions? Meiosis 2, then. What's the difference between meiosis 2 and mitosis? Huh?
Isn't it the same?
Pretty much no difference, right? Except, maybe the chromosomes have crossed over previously, so you're going to deal with a little bit of that, but they're not crossing over in meiosis 2. It already happened. So pretty much no real difference between meiosis 2 and mitosis. So for those of you who finally have a firm grasp on mitosis, then just basically consider meiosis 2 to be mitosis, and it's pretty easy. For those of you who don't have a firm grasp on mitosis yet, you may want to get started. So we're just going to split our chromosome in half, send 1 to 1, 1 to 1, and you end up with each of these, right? Does anybody want to spend like an inordinate amount of time on that since we already have talked about mitosis? Does anybody? I said that in a weird way. I apologize, because now you feel like you can't ask, because then you'll seem like the dumb kid. That's okay. I'm the dumb kid. I shouldn't have said it like that. I don't know. I just don't know what else to say about this. We already talked about mitosis.
So that's the final form of the sperm or the egg?
Yep. So you would end up with one of 4 possibilities. So in this case, I guess it's not trying to say maternal and paternal chromosomes. It's just trying to say like types of chromosomes. So these are all either sperm or egg. And the blue and the red probably just means different kinds of genes, whether that's eye color, hair color, height. I don't know.
Sorry, those are each 23 pairs of?
Each one carries 23 chromosomes. And then when they fuse, they'll make 46. Because we have 46 chromosomes, but in 23 pairs.
And 23 molecules of DNA as well, also?
It would be,
And each
because it's 23 pair, it would be 4. Oh, in each one, it's 23 molecules of DNA. Yeah, because they're single chromosomes. Right here, at least. Each of those contains 1 chromosome and 1 molecule of DNA. Not separately, they're the same thing.
And so does this differ between the males and females?
Does it differ like how? No, not really. They divide the same way, if that's what you mean. You end up with the same numbers, the same products. You just get an egg or a sperm. That's the difference, I guess. I don't know.
So like the sperm and the egg, do they both always contain like, so we have 46 chromosomes, but our halfway cells only have 23. And so is like the sperm always have 1 through 23 and the egg always have like 24 through 46 or something like that? What if there's the chromosome for like personality or something, you got 2 of those and not 1 chromosome for
Then you'd be really annoying. No. It's hard to say because chromosomes don't work that way necessarily, because chromosomes contain insane numbers of genes. So first of all, there's not really like a 1-to-1 ratio. Like your first chromosome doesn't determine hair color, and then the second one doesn't determine eye color, because then we'd only have 23 traits, right? You can't even count how many. So that's kind of a difficult question to answer, but it's hard to say. Basically, there's 23 pairs. And so there's really only 1 through 23. And you get half of the pair 1 from your dad and half of pair 1 from your mom. So they both contain a 1 and then a 2 and then a 3, and then they combine and still just make 1 through 23, but now it's diploid. Does that make sense? But you'd still be annoying if you had too many chromosomes. No. And you can have errors where a sperm contains 24 chromosomes and an egg contains 22 or 24 or 26. I think we get into those a little bit later on. So there are errors that occur with this, but the ideal model is you end up with 23 from mom, 23 from dad. They come together to make 23 pairs. So it's a very long way of answering that. I hope it helps. Other questions?
Can you go over independent assortment?
Yes. We're going to get to it. I think I have 1 or 2 slides from here. I have a slide on it. Should be in there. Is it not? Are you looking at them?
No, I'm not looking.
Okay. You gave me a look and I was like, is it not in there? What happened? I spent so much time on these. Clearly. I like cropped that from Google and just and that's the noise it made and everything. Other questions? No? All right. So this is another way of looking at it, and it kind of helps you visualize how mitosis and meiosis 2 can be the same thing. We take these 4 chromosomes, right? We duplicate them, but we don't turn them into homologous pairs, right? So this one and this one, they don't line up in mitosis, right? Mitosis doesn't care about homologous pairs. They may or may not be the same size. Doesn't matter at all, right? They all line up in the middle. None of them line up with each other. And then they split evenly between 2 cells. Meiosis, on the other hand, does pair them up as homologous pairs. This is where people get tripped up. Homologous pair does not and we're talking about just this picture, does not mean same color, means same size, right? So it's not going to be the same chromosome from the same cell lining up with itself, right? So if this chromosome duplicates, it's not going to line up with itself because A, that wouldn't help. And that's the only reason. I don't know why I said A. So they line up as homologous pairs by size, not by where they came from. Then they line up in metaphase 1, similar to how they line up in metaphase and mitosis, except each homologous pair lines up, right? Then homologous pairs in prophase 1 will do crossing over. That X means crossing over. And so the neighboring pieces will sometimes sort of blend together and then get cut off, and you end up switching bits. And we'll look at that in a second. But they line up in the middle as homologous pairs. Then each half, or each one, goes to one cell or the other, right? Then those get divided in half exactly the same as how mitosis does it, and we end up with 4, right? You guys see how that works? This is just a second chance to look at it. I haven't really told you anything new. Well, I never tell you anything new. We just talk about what happened in class. This is a way to look at the numbers, right? If you're going, I just don't see how mitosis divides stuff in half, right? Because we divide our chromosomes here, but we still have, well, let's look at mitosis first. So mitosis takes and splits the it doubles the chromosomes and then splits them in half. But if we double and then split in half, what do we have? Same as what we started with, okay? Now, diploid, mitosis does what? Puts them homologous pairs, right? Then splits those in half without duplicating, and that's how you end up with half as much as what you started with. Right, 23 rather than 46 chromosomes. Does everybody see how these numbers are coming about? Anybody not see how the numbers are coming about? You afraid to be the dumb kid still? That's me too. I take Ochem this semester. I have so many questions the whole time, but I just sit in the back and I'm like, I don't want to be the only one that doesn't know how to ask. So if you don't want to be the only one that doesn't want to ask questions, email me, come up to me afterwards. In fact, people may have been still emailing me like it's the super session. Nope, just Utah State Athletics to let me know it's homecoming week. So that's fine. You can email me during this session. You can email me during any session. Sorry.
So we go back one slide.
No.
Okay, so it has the diploid. This is in meiosis. It has the 46 chromosomes. When it splits it into the 2, do they also have 46 chromosomes?
Somebody want to help me? Let's do some math here.
And then it goes into 23.
Then it goes into 23, but who wants to help me? How many do we have here? Oh.
Each cell has 46, but it's haploid, so it's not diploid.
Now you got me confused. I don't know the answer. All right, so let's go back. That's actually a very valid question. Let's reason this out. So you're asking, let's compare these. So this is the start, this is the end, and this is the middle. So you're asking from here to here, how many chromosomes does each cell have? Right? Help me out, guys.
23.
One person says 23. Does anyone disagree? One person disagrees. So we're going to have a fight night to the death, and the winner is right. That's how science is determined. Whoever's the strongest. Okay, we got some chatter going on. Quiet down. We got questions and or comments.
Well, it would be 46 because you still have the pairs. Because in meiosis 1, you have the 2 pairs, but when it splits, you still have the homologous. Am I right?
So
46, and then that's when they split to be 23. But what I don't understand is why don't they start out as 92 when it's already been, and then break it down to 46 and
I see what you're saying. So we have our homologous pairs lined up, right?
Yes.
So you're saying you're not sure why that's not 96?
When they say it starts at 46.
'Cause it's
what
46.
We're all getting confused is because we forget that there's still 2 duplicated chromosomes when we split down
Right.
before that.
Right. So it's 96 DNA molecules, but because you're dealing with chromosomes, 2. What?
You mean 92?
92. I don't know math. Don't ask. No, sorry. So the chromosomes are made up of 2 DNA molecules. We have 46 chromosomes, 92 DNA molecules. So this is dealing with chromosomes, right? Maybe that'll help. So we have our homologous pairs, right? Maybe this one looks a little bit better. We have our homologous pairs, right? And how many chromosomes do I have here? 2. How many chromosomes do I have here? How many chromosomes do I have here? So, how many chromosomes do I have here? How many chromosomes do I have here? 46, right? The homologous pairs have separated, but the number of chromosomes hasn't yet been split in half because the chromosomes haven't split in half.
Homologous pairs before separation only count as 1 chromosome.
Say a little louder, I'm wearing a beanie.
Do homologous pairs before separation only count as 1 chromosome?
No, they still count as, well, each chromosome is a chromosome. That's a terrible way to explain that.
So when you split
Each one's a chromosome, even as so the homologous pairs are each a chromosome, right? I'm making this worse, I swear. I'm trying to think of a way to not make this worse. I don't know if drawing it out's going to make it any better. I feel like this is going to be the best way to look at this. So, guys, each chromosome duplicates, right? And then it lines up with its homologue, right, making them a homologous pair, right? Maybe I have got this backwards. Hang on. What? I may have this backwards. You sound like you know what you're talking about. No. As you say that, I think I see where I went wrong. So they line up, right? And I falsely said that that's 2 chromosomes. That's 4 chromosomes right there, right? Because that's a chromosome and that's a chromosome. I made it sound like they were chromatids, right? Each of those is a chromosome made up of 2 chromatids. It's also kind of, the picture's a little wacky. So how many chromosomes is that? Just what I circled. 2, right? And another 2, okay? Now, how many chromosomes is that? 2. So we went from 4 to 2, okay? So whoever's in that corner sitting on the ground there's like 15 open spots, by the way. If the floor's more comfortable, you do as you please. The chromosome number, so let's say chromosome number goes down in meiosis 1. DNA molecule number goes down in meiosis 2. So this is how many chromosomes? 2. This is how many chromosomes? 2. How many molecules of DNA? 2 and 2. Okay? So chromosome number goes down in meiosis 1. Number of DNA molecules goes down because each chromosome gets split in half. It's still the same number of chromosomes, but now it's half as many DNA molecules.
So if we were to go back to the other example, it would be 23 after meiosis 1, right?
Yeah, so this should have been 23 chromosomes still 46 DNA molecules, followed by 23 and 23 chromosomes and DNA molecules. So I'm blaming the picture, when in reality, I just said it wrong. Let's be real. And I do that sometimes. So is everybody clear now? No. I'll take that as a no.
So the 23, and then you're just replicating those last four outputs, right?
Not so much replicating, but these 23 are these kind of chromosomes. These 23 are that kind of chromosome. So I take and I split that chromosome in half. It's still the same number of chromosomes. That's one chromosome and that's one chromosome, but now it's half as many DNA molecules. Make sense? Okay. I finally answered one right, you guys. Thank you.
So,
And no one was paying attention. They're like, why are we... What are you guys doing? Quiet down. She has a question.
okay, so in the middle one, so there's 46 DNA molecules.
This has, does anybody know, 46 what? Chromosomes. How many DNA molecules? 92. Because these are duplicated chromosomes. So they have twice as many, they have 2 DNA molecules per chromosome. Does that answer that?
Yeah.
So meiosis 1, we're reducing chromosome number, right? Meiosis 2, we're reducing DNA molecule number, because this is 23 and 46. Isn't genetics the worst? Got a couple of questions.
So where there's the empty spot that we just figured out was 23, right?
Yes.
So at the end of meiosis 1 and then the end of meiosis 2 is with the 423.
Yeah, so this whole process is meiosis 1, and then from there to there is meiosis 2. Yeah? Do you have one?
So would you say that in meiosis 1, both the chromosome number and the DNA molecule was reduced by half, and then only the DNA molecule was reduced by half?
True, because as you split up the homologous pairs to each cell, you are also dividing the amount of DNA molecules into different cells. Yeah, that's a valid point. So I guess they both get reduced. Did everybody hear that? That was actually it's a valid question. No? We got like 18 side conversations going on. Come on, guys. He asked, so there's 46 chromosomes and 92 DNA molecules. Then there's 23 chromosomes and 46 molecules. Didn't we reduce chromosome number and molecule number? Yes. Why? Well, we didn't cut anything in half.
The pairs got separated.
The pairs got separated. So how many DNA molecules is that? 4 DNA molecules. How many chromosomes is that? 2. If I split those up, how many chromosomes in that cell? 1. How many DNA molecules? 2. So we cut in half the number of chromosomes and DNA molecules from the start to the end of meiosis 1. Then what do we cut in half? Not chromosome number, because we go from 23 to 23. Molecule number, we go from 46 to 23. Because we take this chromosome and we turn it into those chromosomes. And this is per cell. We have 1 chromosome here, 1 chromosome here, 1 chromosome here. But we have 2 DNA molecules here and 1 DNA molecule there. Okay? This is something that is a somewhat tricky topic, so it's awful that he decided to do this right before the test. I know it's terrible. Hopefully you signed up for Thursday. If not, you may want to reconsider. Go over this a bunch, right? Unfortunately, I have 10 minutes left in the session. I can't allocate any more time to this. I know that's the worst. We still got a lot more stuff to cover, okay? So go home and study this, okay? Watch this lecture again, or if I confused the crap out of you, which I probably did, watch Dr. Bogorski's lectures, because he's got a PhD in this stuff and is a million times smarter than me. So, as promised, we're going to talk about independent assortment, right? Independent assortment is a type of genetic variation, right? They can line up on either side of each other. So either you can have these two like this and like that, or you can have the top two are still in the same place, but these bottom two, do you see how they've kind of flip-flopped, right? So the red one's now on the left and the blue one's on the bottom, or the blue one's on the right, on the bottom two. Why didn't the top two do it also? Huh? Well, kind of. You're saying it's chance whether the top one or the bottom one does it. That's true. It's 50-50 chance that the top two or the bottom two switch. Why didn't they both switch at the same time?
If they both switch, it's the same thing.
Yeah, it would be the same thing. You would just be looking at it like backwards, right? So they'd still end up all blue in one side and all red in one side, right? So you can either end up with all blue and all red, which is going to lead to all blue, all red, or you can end up with blue, red, red, blue, so you get blue, red, red, blue, right? So that's one way that's one of the reasons that we don't all look like each other, right? Not all of our chromosomes lined up in the same orientation when they split in half. So some of us ended up taller or shorter or brown hair, blonde hair, red hair, whatever, right? Is anybody not feeling independent assortment?
Well, what would limit independent assortment?
It-
There's a question that said like a haploid cell is looking to, trying to multiply.
Mhm.
It's talking about crossing over versus independent assortment. So what would limit independent assortment? Instead of haploid couldn't, but why would that be?
Because it has to have a pair.
It has to be more than
See how we wouldn't be able to switch pairs if there's not a pair? Because you can put one chromosome on the left or the right, but it's not compared to anything else. So you're not switching it because it's not switched against anything else. The question is, why can't haploid cells do independent assortment? Because they only have one copy of each chromosome. So if they're all lined up in the middle, right, you can't really put one to the left or one to the right because they're all in a straight line. But if I have two, then I can flip-flop those, right? So that's why that's one of the limitations of independent assortment. It only works in diploid cells, and it only works as a 50-50 chance, right? So that's not a whole lot of genetic diversity when you consider thousands of traits out there, tens of thousands of traits. Just flipping them one side to the other only gives you twice as many traits. So if this were the only way to get genetic diversity, there would only be two traits. You would either only have black hair or blonde hair, and that would be it, right? Because you would only be able to line up one of two ways. So that's not true because of crossing over. So this is what I talked about. It happens in prophase 1. Where's prophase 1? Meiosis 1, right? So these homologous pairs line up. They do something called chiasma. See how they're like pushing up against each other? I guess red on red doesn't work. But they literally touch each other and cross portions and then get cut off at those portions, and they switch sides. They literally like dark side, light side, trade sides, right?
I had a quick question on the previous slide. So when in humans, is that happening with all 23 different chromosomes? And so that's part of what gets us so many, I don't know, possibilities?
Yes.
Okay.
So it happens with all 23 chromosomes. Each one can line up in a different way. So it'd be like 2 to the 23rd possibilities. I don't know how to do probability. It's a lot of possibilities. But again, it's not that many considering how many trillions of people and trillions of traits there are. But it is a lot. It's just not as much as crossing over.
Will you have either like independent assortment or crossing over, or do you have both?
Most of the time you'll see both. Crossing over happens less frequently than independent assortment, but a lot of times both will happen. So they'll line up, they'll assort independently, and then they'll cross over. Does that make sense? So a lot of times it's both. So they touch each other, and then arms get cut off and exchanged, right? And they trade sides. So that's how you end up with this red-blue chromosome, right? See how that is vastly different from that? Because it's made up of all kinds of pieces. And you can end up with different lengths. You could end up with like that much red and only that much blue. And this has nearly infinite numbers of possibilities. That's why literally everybody except for identical twins looks different. Siblings even look different. Because their chromosomes crossed over different from their siblings, right? There's so many ways crossing over can change things.
So is this with like our own? Is this with duplicated chromosomes? I guess I just don't understand how this creates like diversity when it's with, the same.
So let's say this blue chromosome is from mom and the red chromosome's from dad, right? So let's say your dad has black hair and blue eyes and is really tall, right? And your mom has blonde hair and green eyes and is really short, right? You, without crossing over or anything like that, you're either going to end up tall, black hair, blue eyes, or short, what did I say? Blonde hair, green eyes, whatever I said, right? If you cross them over, you can get brown hair but green eyes but short, or you can get tall but blue eyes but blonde hair. So you can mix stuff around so that your genes from mom and dad kind of become a hodgepodge. That's why you aren't like half mom, half dad. You're just all of it.
So is this like only in fertilized cells, or like do our cells know which DNA came from?
No, they don't. Cells don't keep track of where anything came from. They don't say, we'd rather take the egg's DNA or the sperm's DNA. And the eggs don't say, that sperm seems better, that sperm seems worse. It's whoever's fastest, right? Whichever sperm gets there first out of thousands, tens of thousands, right? So that's another thing that's random, is random fertilization, but I'll get to that in a second. But this is just in your zygotes, right, in the egg and sperm, they're mixing up and going however they want. And when they meet up, they mix up and they do whatever they want.
So this is only in zygotes and fertilized cells?
Yeah. This is because it has to be your sexually reproducing cells. I think like regular cells can do this as well, but it's pretty rare since they don't do meiosis. So it's pretty hard for something like this to happen in a regular cell. So it can only happen in meiotically dividing cells, and the only cells that divide meiotically are zygotes, right? Does anyone disagree with me? I hope not, because in that one I'm actually right. All right, so random fertilization is just the idea that any sperm can fuse with any egg, right? Men will produce trillions of sperm in their lifetime. Women will produce tens of thousands of eggs in their lifetime, right? And in every fertilization event, there will be thousands, tens of thousands of sperm, okay? There's usually only one egg available at the time, but it's not, they don't come out like in order, right? They don't descend into the uterus in any specific order. Any egg will just, I don't know, that egg's just the one that comes down the fallopian tube into the uterus, right? Nobody's up there saying, that's number 3, and that's number 4, and like we've been keeping really good track. No, they're just up in your ovaries, doing whatever, who knows, nobody keeps track. And one of them just comes out, right? And it's completely different, well, not complete, it's pretty different from the rest of the eggs, right? So depending on which egg happens to come out and which of the tens of thousands of sperm happens to be the one that fertilizes it, that's the genes you end up with. And that's why your siblings will look different, partly crossing over, partly also, they're not from the same egg and sperm unless they're your identical twin. Then they look exactly like you. So that also explains why they look exactly the same. They got the same genes, right? Does that make sense? Do you guys see the 3 kinds of genetic diversity there? How they operate? Because you got a test on it technically tomorrow. All right, the worst part, actually this really isn't the worst part. I only left a minute for this because in reality, all you have to do is draw this a bunch of times until you have it memorized, and know that animals spend most of their time diploid. Fungi and some algae spend most of their time doing what? Or as what? Haploid. And then plants and other algae, spend most of their time as, it's even, right? So animals spend most of their time as diploid, fungi and some algae spend most of their time as haploid, and then plants and the rest of the algae spend an equal amount of time as diploid and haploid, okay? That's all you need to know there, plus draw those, okay? Draw them a bunch of times until you're able to put the right stuff in the right order with the right ends, okay? We're out of time for today. Oh, I forgot to pass around the roll, didn't I? That's not a good way to do it. You guys get a freebie today. There's no roll. It was sitting right here the whole time.